The small intestine has a lot of things going on for it. It's got a lot of neat, amazing things. We just recently discussed the liver and the gallbladder, as well as the pancreas, and how they play their roles in the small intestine. If you haven't watched that already, I would suggest doing that first before moving on. But then again, it's not super extremely vital important that we do. The small intestine can be broken down into three parts: the duodenum, jejunum, and ileum. The duodenum being the shortest of it all, maybe 6 to 12 inches, the jejunum several feet long, with the ileum being the second longest, and it's also the latter portion of the small intestine. Knowing this order is vital and essential. The small intestine has a lot of things going for it. It's the place where you're going to see 90% of absorption occurring. The stomach, as well as the liver, pancreas, as well as the many enzymes embedded in the small intestine as well, have broken down these molecules into the smallest components possible. That way absorption can occur. The layout, the inside lumen of the small intestine has everything necessary to make it be able to absorb as much as possible. How it does it and why I say it that way is because it has folds on folds and folds. The inside of your small intestine, by increasing its surface area, also increases the number of chances it's going to find food particles, the chyme particles, and help digest it further if necessary, or absorb it if it's small enough. And I say folds on folds, we get to see these macroscopic folds in a partially dissected small intestine, but if you get nice and close, you're going to see that those folds even have more folds on top of them. If you were to take a closer look at your slides, you'll see that there are villi on top of each individual of these folds. And then upon these folds, these villi, there's the circular folds, there's the villi, you'll find on top of them villi of the cells themselves. Those, you literally have folds on folds. The old meme is coming up and says, hey, I heard you like folds, so I gave you folds on your folds, type of thing. Different types of cell types again. You got the mucosa cells, the absorptive cells. The mucosa cells, I should say, more along the lines of goblet cells. If you remember back from A&P3 when we talked about the lining of the lungs, there were those goblet cells that produce mucus. Well, these goblet cells here also produce mucus. You got absorptive cells that are there to help absorb things. You got the lacteal cells and some enteroendocrine cells also associated with these. Do note that hopefully this should sound familiar. You got arteries coming in, delivering oxygen, and because you're in the small intestine, you've caused particulates, nutrients to come across. They get absorbed into these capillary beds and brought forth into the venules and eventually into the veins and all that stuff to get up to the hepatic portal system. Hopefully you'll also remember that we have little lymphatic systems here. This is the special name for it is called a lacteal. The lacteals is a branch of that lymphatic system, and if you remember, fats have to be absorbed via the lymphatic system. They do not go into the blood. They have to go into the lymphatic system first. Several cross sections of these tissues. There's that circular fold again. You'll see villi there. Here's a electron scan microscope picture of it so you can see those villi a little bit better. So microvilli, circular folds upon villi upon microvilli. Now, there are two different types of movements that this small intestine can do. It's segmentation and peristalsis. Segmentation, if you remember, you don't is helping you churn the food. In your mouth, you got teeth and a tongue to help churn and move and manipulate that food, but segmentation is there to help mix and churn that food. It doesn't mean that you're moving food directly down the small intestine all the time. It also means that you're mixing it back and forth, back and forth. One reason to help um, get those nutrients to that cell membrane, as well as to emulsify those fats a little bit better, mix those bile salts a little better, as well as mix those enzymes in really nicely into that food so they can find, or that chyme, so they can find those molecules and break them down further. The movement of actually moving things, the action of actually moving food along the small intestine and towards the back end of your body is called peristalsis. Peristalsis, if you remember back in our previous movement talking of the muscularis layers, you got the two layers, the circular and longitudinal. The circular doing, in this case, are doing the segmentation mostly, while the longitudinal having a role in that peristalsis. Peristalsis meaning that you're moving that chyme along through that tube and towards the end of that tube. There is chemical digestion in the small intestine. We already talked a little bit about that in the pancreatic, the brush border enzymes, the maltase, sucrase, lactase, the things that are embedded into that plasma membrane. I'm not going to go into huge detail here because there was in the liver and pancreatic discussions. But what I'm going to go ahead is talk about the absorption. We have broken down everything. We have broken down those macromolecules into monosaccharides. We've broken them down into amino acids or di or tripeptides. We've broken down the nucleic stuff that's not really present here, but it is. We've broken it down to the point that we can actually absorb it. We've also broken down those fatty acids from triglycerides and long chain fatty acids down into monoglycerides and large short chains and somewhat long chain fatty acids. They are now ready to be absorbed. Depending upon their characteristics, the molecular characteristics will determine how they are absorbed. For instance, fats. Those plasma membranes of your cells will absorb those fats quite nicely and readily via simple diffusion. Once they have absorbed it, they will repackage those monoglycerides into triglycerides into chylomicrons, which will then eventually enter into those lacteals, into that thoracic duct, into your lymphatic tissues, and on up to that left subclavian. You'll also notice this is actually, I just realized this has been a little while since we've discussed diffusion and all that stuff and the varying types of transporting things across the plasma membrane, so I should probably hit up quite a few of these things. For instance, facilitated diffusion is just a really fancy way of saying there's a protein that has made a hole that allows that, whether it be gated or not, that allows these things to easily just pass on through with its concentration gradient. So fructose is one of those that's going to pass through that way. The other ones have to pass through via an active transport. They have to use some type of force to be absorbed. Secondary active transport, you're using a concentration gradient. For instance, glucose and galactose. You have, if you're remembering back from the pancreas is releasing a bunch of sodium into that. Well, we'd want to recover and recoup that sodium. So we're going to use that concentration gradient to absorb that sodium in. And since that's already happening, why not use that power to draw in glucose and galactose? You're also going to do that in parts for a few of the amino acids. There are several amino acids, I believe there's about 21 now, that you need to absorb. Some of them are done via secondary active transport using that same sodium ion. You're also going to try and reabsorb these dipeptides, tripeptides in a secondary active transport type of fashion, but you're going to use that hydrogen ion concentration to drive that movement of those dipeptides and tripeptides in. Active transport is referring to you are using literal energy. You're not just using electric energy. You're using adenosine triphosphate, ATP, to make that thing move. These cells are dedicating ATP to draw in those good portion of those amino acids. It's also using ATP. Well, that's the only one that's really using that active transport. Again, Everything is absorbed into the blood minus the fatty acids and the triglycerides and all that. They're absorbed as chylomicrons and sent on down the lymph. The rest are doomed to go and see that hepatic portal system be sorted, used up, or modified by that liver. With that, that is the end of this discussion of the small intestine. Remember, some major points are that it absorbs about 90% of your materials and contents. I'm going to go ahead and point out here before I move on that the large intestine takes about 9.9 to 10% of those nutrients up as well, particularly in the form of salts and water. And the liver, not the liver, the stomach has a very, 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 very small portion percentage that it absorbs. Usually it's just things like water and alcohol. With that, good luck.